are starting to our class. That is also an important class, whatever we have understood into the previous class. I always say you that just subscribe our channel so that you can get notified in every kinds of classes. Today's topic going to be very much interesting, that is the Salmonella. We are going to understand the Salmonella within a very short time. Just stay tuned with us so that you can easily take the idea regarding Salmonella. See, just subscribe our channel for daily classes and just check our description box for lots of free content you may use this. See, first one is the Salmonella. We need to understand that we have already understood into the previous classes that how our methodology to understanding each bacteria. If you are understanding one bacteria's methodology, how the study properties as well as how the study way, you can automatically understand every kinds of bacteria. See here, firstly we need to understand about the introduction. See, introduction means we need to understand that what is basically Salmonella. See, Salmonella is a gram negative first thing. Second thing, it should be the bacilli shape because they all are the rod defining the bacilli. Got it? And third one is they are facultative anaerobic. They defining that or these kinds of bacteria can easily grow without oxygen and they can also grow with oxygen. Okay, as well as they are belonging into the Enterobacteriaceae. There have another type of bacteria. They all are belonging into the Enterobacteriaceae. See, all the gram negative bacilli, almost gram negative bacilli are belonging into the Enterobacteriaceae, like Escherichia coli, like Salmonella, like Shigella. They all are belonging into this family. See here, they basically, the Salmonella names basically coming from the Daniel Ilmar salmon. Okay, there have basically two types of species. First one is the Salmonella enterica, another one is the Salmonella bongori. Okay, Salmonella enterica is mostly prominent as well as clinically important because you might lots of time listen that Salmonella typhi, Salmonella paratyphi, they all are under the Salmonella enterica. Got it? See, here is a genus, that is the Salmonella. If I am coming to the species, that is the Salmonella enterica. If I am coming to the subspecies, here I have also subspecies. See, that is the enterica. They all are most pathogenic strains. Got it? And without the Salmonella enterica, there have also two types. See here, first one is the Salmonella typhi and another one is the Salmonella paratyphi. We all know that about the typhoid fever, right? That is the waterborne fever basically spreaded, and this is caused by the Salmonella typhi. Salmonella typhi is the most prominent. Got it? And here, see here, that is a rod-shaped bacilli. That is the Salmonella. See here, rod-shaped bacilli. Understood? See, now I am coming to the second slide. It's also important. See, we are getting to understand that as well as we have to take the idea regarding how the Salmonella look like. See, first one is the shape. If I am coming to the shape, that is a rod shape. I have already told you that after the gram reaction, they all are the gram negative. After that motility, they all are the motile because they have the peritracheous flagella. I always say you that the methodology, how do you understand each kind of bacteria? Just check out our previous classes. You all can easily take the idea of every kinds of bacteria. Every kinds of bacteria reading methodology or understanding methodology is completely same. Got it? Here is a peritracheous flagella. Peritracheous flagella defining that the flagella should be present all over the surroundings of the bacteria. That is called peritracheous flagella. Okay, peri defining that all over the bacteria or all over the surrounding the bacteria there should be present flagella. So that it's peritracheous. After that, they all are the non-sporing. They cannot grow as well as they cannot form any kinds of spore. Okay, and there should be capsule present. VI antigen or Salmonella typhi enhance the virulence. The Salmonella typhi basically virulency become increased by the Salmonella typhi. Got it? Now if I am coming here the structure, see here that is the basic structure. They all are the flagella. Surroundings the bacteria they basically present, so that we are saying that erythrocytes, okay, and there should be present the DNA, there should be present the plasmid, and the outermost layer that is the cell wall, and complete outermost layer that is the capsule. Here is a VI capsule is most prominent. Understood? Now I am coming to the antigenic structure. There are basically three types of antigen. First one is the O antigen, that is the somatic, or we all know that somatic antigen basically present outside of the cell membrane. Understood? After that there have the H antigen. H antigen always the flagellar antigen, so that this antigen should be present into the flagella, that is the H antigen. Got it? See, after that VI antigen, they basically present into the capsule. They are founded into the Salmonella typhi and Salmonella paratyphi, increase the virulence. I have already told you that. Got it? And Salmonella as O antigen or somatic antigen is the heat stable, means they can easily tolerate the heat, and the Salmonella's H antigen, that is the heat labile antigen. See, that is the heat labile antigen, they cannot tolerate the heat. Got it? That's all are the antigenic variation. Okay. Now I am coming to the cultural characteristics. We need to understand that in which media we can use this for the Salmonella. See, they are aerobic, means they can easily grow with oxygen, and they are facultative anaerobic, means without oxygen they can also grow. Without oxygen they can also grow, and with oxygen they can also grow. Got it? See, now I am coming to the cultural characteristics. Though whenever we are discussing about any kinds of cultural characteristics, we need to understand that there have lots of cultured media, okay, but we are understanding only few, so that we can easily memorize on the aspect of the Salmonella. See, first one is the nutrient agar. They can easily grow into the nutrient agar. They moist and translucent colonies. Got it? After that, see here McConkey agar. They should not ferment the lactose, so that we will see that colorless colony, non-lactose fermentation. Okay. After that, they can easily grow into the XLD agar. Into the XLD agar, they are basically founded the red colonies with black center, and black center are formed as well as the black center formations are happening due to the estrus production. Okay. And another one agar there is also present, that is the hectone, and there is a hectone and SS agar. Okay, green or colorless colony might be formed, and you need to understand that, as well as you need to memorize that the temperature should be 37 degrees centigrade. Got it? There is also one cultured media's example basically given in every slide. You find out the picture, so that you can visualize what I am talking, as well as what's I wanna teach you. Understood? See, after that we are coming to the biochemical test. Please keep in mind that we have already understood the morphology. We have already understood the morphology of Salmonella. We have already understood the morphology. We have already understood. After that, we have already understood the cultural characteristics. Now it's a high time to discuss about the biochemical test. See here, if I am just writing here, see, biochemical test means you need to understand which kinds of tests are positive and which kinds of tests are negative. Firstly, see here, Salmonella basically catalase positive. Okay, they basically produce the bubbles. Okay, catalase positive means there should be H2O2 become converted to H2O and O. Okay, after that, see here, oxidase positive, oxidase negative. Got it? Lactose negative. After that, indole negative. Okay, MR methyl red, that is positive, BP negative. Okay, citrate, that is citrate positive, urease completely negative. Got it? Motility, I have already told you that they become motile. Okay, they become motile. After the TSI tri sugar iron or triple sugar iron, they become positive. Okay, and you will understand the particular things whenever you will attend the class of biochemical. Okay, biochemical test, they all are the general microbiology part. If you are understanding the biochemical test at first, before understanding the systemic microbiology, you can automatically understand it properly. Okay, we have already covered the biochemical test. Now we are coming to the disease condition. Which kinds of disease might be occurred? We need to understand it. First one is the, see here, that is a typhoid. We all know that typhoid are caused by the Salmonella typhi and Salmonella paratyphi. We all know that. After that, within the typhoid, basically high fever occurring, rose spots occurring, hepatosplenomegaly might be occurred. Okay, after that, gastroenteritis, that is also happening by the Salmonella. Another species like Salmonella enteritis, like Salmonella typhimurium, okay, they causes the diarrhea, vomiting, cramps. Okay, and bacteremia might be occurred. Bacteremia defining that whenever the bacteria are present into the whole over the body, and sepsis whenever the bacteria become grow within the whole body, that's called sepsis. Okay, that might be occurred, as well as localized infection might be occurred. That's all are the disease condition. And after completing the class, if you want to take the PPT, then please join our free group. You will get the PPT daily basis, in which day class will be happen, and you will get the PPT. And into our free group, there we are providing the 3 classes per week, that is completely free of cost. Okay, 2 class you will get in YouTube, and 1 class you have to join into our free group. You will get the notification of the free classes. Okay, not only classes, but also you will get lots of benefit from our free group. Okay, like one liner, like tables, like notes, like MCQ, like mock test, like vacancies, like M.L.T, all kinds of solution you will get. Okay, that's completely free of cost. That's completely free of cost to all M.L.T students and professionals. Okay, see, here is also one symptoms basically present, as well as here is also the disease condition might be occurred like that. See, there is a rashes into the body, got it? There is a ulcers into the intestine might be occurred, splenomegaly, spleen enlarged, hepatomegaly, liver enlarged, there is a white coating on the tongue. They all are the symptoms of the typhoid, as well as they all are the symptoms of the Salmonella fever, as well as the enteric fever. We all know that, right? Because they all are the caused by the Salmonella enterica, or they all are the caused by the Salmonella typhi and paratyphi. Got it? Now it's we are coming into the last stage, that is a completely last stage. We need to understand the laboratory diagnosis. Though we have already covered that culture, we have already covered the biochemical test, we have already covered the staining. See, that is the, we have to collect the blood, urine, as well as stool. We may collect it. After that, we may do the blood culture, positive in first week, stool culture, positive in the later stage, as well as media. We may use the McConkey, XLD, SS agar. I have already told you that which kinds of media you can use for the Salmonella. After that, I am coming to the serology. Within the serology, we may do the viral test. Into the viral test, Salmonella basically gives the O and H antigen positive. Okay, please keep in mind that within the viral test, we may detect the Salmonella typhi and Salmonella paratyphi. H and O antigen of the Salmonella, we may detect it. Okay, and nowadays that's basically too much uses, because that is a one type of rapid test as well. Okay, now it is too much uses, as well as prominently uses into various laboratory. ELISA, that is also one type of prominent diagnosis. PCR, we may use this for identify the nucleic acid. Got it? And biochemical ID, I have already told you that TSI, we may do citrate test, we may do indole test, we may do urease test, we may do ETC, means I have already covered that. So that within the lab diagnosis, first we have to understand the specimen, after that we have to understand the culture, after that we have to understand the serological test, after that we have to understand the biochemical test. Got it? Now I am coming to the treatment portion. See, for the typhoid fever, there have also ciprofloxacin, ceftriaxone, after that azithromycin. Though the treatment is not our part, there is a suggested treatment that is not, please don't apply into the practical life, just go to the doctor will suggest you the proper treatment. But into the microbiology aspect, we are just providing the basic treatment as well as basic drug of the particular disease. Got it? Into the MDR cases, means multi-drug resistant cases, you will go to the azithromycin, after that carbapenem, okay, into the gastroenteritis. You may uses the self-limiting, usually self-limiting antibodies, okay, and last but not the least, that is the bacteremia. Into the bacteremia condition, you may uses the third generation cephalosporin, okay. That's all are the treatment portion. Okay, thank you for joining. Just subscribe our channel for regular classes and weekly 3 class you will get, and please join, join, as well as I am just requesting all of M.L.T fellow, not only our benefit but also your benefit. Please, you may join our M.L.T free group. Okay, we have our free group. We are providing notes, we are providing MCQ, we are providing mock test, we are providing complete vacancies notification, we are providing classes, everything completely free of cost. Okay, we are providing ebook as well. Everything is completely free of cost. Just join our group. If you are not getting the benefit, then you may left. Okay, that's all we are doing only for the M.L.T students, so that our platform is only for the ultimate M.L.T solution. Okay, thank you so much for joining. Hope we will meet into the next class.